So today, we're going to trace the incredible, fast-paced life story of a single cell. It's a journey of growth, replication, and division that we call the cell cycle. Have you ever really thought about how a cut on your finger just heals? Or how you grew from a tiny baby into, well, you? It all comes down to this absolutely beautiful, complex process that's happening inside your body right now, millions of times every single second. It's the life story of your cells. So what exactly is the cell cycle? You can think of it as a cell's entire life, from the moment it's born to the moment it divides into two new daughter cells. And the goal? The goal is simple, but it's absolutely vital. Make two perfect identical copies. Now, a cell's life is basically split into two major parts. The first one, and by far the longest part, is called inner phase. This is the prep stage. A cell actually spends about 90% of its life right here, just getting ready for the big event. Honestly, the best way to think about interphase is like a training montage in a movie. You know the one? This is where our cell is bulking up, it's gathering resources, and most importantly, it's making a perfect copy of its most critical possession, its DNA. And this training montage has three acts. First up, in the G1 phase, the cell just grows and stockpiles energy and all the building blocks it needs. Then comes the really crucial part, the S phase. S stands for synthesis, and that's where it copies all of its DNA. And finally, you've got the G2 phase, which is for some last-minute growth and one final check to make sure everything, and I mean everything, is ready to go. During that S phase, the cell's DNA gets meticulously duplicated. Each chromosome now has an identical copy of itself, and these two copies are called sister chromatids. They're literally joined at the hip. The cell has officially photocopied its entire genetic library. Okay, with all the preparations complete, the cell is finally ready for the main event. This is the mitotic phase, and it kicks off with a spectacular process of nuclear division called mitosis. This is where all that duplicated DNA gets sorted out. The best analogy for mitosis is a dance. It is a carefully choreographed series of precise, organized movements, and it's all designed to do one thing: perfectly separate those sister chromatids so that each new cell gets a complete and identical set of genetic instructions. No mistakes allowed. So this dance has four main acts: prophase, metaphase, anaphase, and telophase. Each one is a totally distinct step in the process of dividing up the nucleus and its precious cargo. Act 1 is prophase. Here, those long, stringy strands of DNA coil up super tightly. They condense into those classic X-shaped chromosomes you've probably seen in textbooks. At the same time, the membrane around the nucleus starts to break down, basically setting the chromosomes free. Next up is metaphase. Meta means middle, and that is exactly what happens. It's pretty amazing. All of the duplicated chromosomes are grabbed by these microscopic fibers, and they're pulled into a perfect single-file line right down the equator of the cell. Now for the most dramatic part of the dance, anaphase. The sister chromatids are suddenly ripped apart from each other. Each one, which is now considered its own full chromosome, gets dragged toward opposite ends of the cell. It's like a game of tug-of-war where everybody wins. And the dance concludes with telophase. The chromosomes arrive at their destinations at opposite poles of the now elongated cell. The cell then basically hits rewind on prophase. New nuclear membranes form around each set of chromosomes, and suddenly you have two distinct nuclei inside one big cell. So now we have two complete sets of genetic instructions, each in its own nucleus, but it's still one big cell, right? The final step is to divide the rest of the stuff, the cytoplasm. And this part of the process is called cytokinesis. And it turns out animal and plant cells handle this a little differently. In animal cells, our ring of proteins basically acts like a drawstring, cinching the middle and pinching the cell in two. But plant cells have that rigid cell wall, so they can't just pinch. Instead, they build a new partition called a cell plate right down the middle, which eventually forms a new cell wall. This whole process is just unbelievably complex, and it has to be perfect. A single mistake could be catastrophic, which is why the cell cycle doesn't just run on autopilot. No way. It's governed by a very strict set of rules and quality control checks. So how does the cell make sure it doesn't copy damaged DNA or divide unevenly? How does it maintain this incredible precision millions and millions of times over? Well, the answer lies in a series of internal checkpoints. Think of these checkpoints like molecular security gates. There are three main ones. You have the G1 checkpoint right before the cell copies its DNA. There's the G2 checkpoint right before mitosis begins. And there's the M checkpoint right in the middle of mitosis. At each one, the cell pauses and asks a critical question before it gets the green light to proceed. So at that first gate, the G1 checkpoint, the cell asks, am I big enough? Do I have enough energy? Is my DNA undamaged? If yes, it can move on to the S phase. Then at the G2 checkpoint, it does a quality control check. Was the DNA replication successful and error-free? Finally, the M checkpoint stops everything mid-GANs to confirm that every single chromosome is properly attached to the spindle fibers before they get pulled apart. Now, not all cells are constantly on this cycle. In fact, many of our most important cells, like mature nerve cells or heart muscle cells, actually exit the cycle. They enter a resting state called G0. They're still doing their jobs, of course, they're just not actively preparing to divide. And this quote from our source material really drives home why all these rules are so important. It says, it is essential that daughter cells be exact duplicates of the parent cell. Mistakes lead to mutations. The cell cycle's #1 job is to prevent that from happening by ensuring absolute perfection from one generation to the next. Which brings us to one last really crucial question. We've seen this elegant dance. We've seen the strict rules that govern a cell's life. But what happens when a cell goes rogue? What are the consequences when it starts to ignore those checkpoints and just divides and divides and divides, completely out of control?